Good morning. In this video, we will pursue our discussion of the chloroplast and its role in photosynthesis and how it is adapted to carry out the function of photosynthesis. As you know, the chloroplast is one of the organelles that are surrounded by a double membrane, an outer membrane that is rather smooth and an inner membrane that is folded. And the folds of the chloroplast are um, more extended than the folds of the mitochondrion, of the inner membrane of the mitochondrion, and they are folded into what we call the thylakoid membranes. So here, as you can see, the inner folds of the chloroplast are thrown into projections or folds that form like membranous sacs that are piled one over the other, and each of them is called the thylakoid, and the pile of them is called the granum, and in between them we have an extension of the membrane that joins the grana together, and this we call the lamella. And the importance of the lamella is to increase the surface area and therefore increase the content of the pigment inside the chloroplast. And here you can see that the outer membrane is smooth, that the inner membrane, this one, is the one that is folded into thylakoids and grana. And in between them, we have a space that is called the intermembranous space. So here we have, inside the chloroplast, we have piles of thylakoids. And as we said before, the thylakoids are folds of the inner membrane of the chloroplast that take the shape of discs that contain the pigment, and they're called the thylakoids, and a pile of them is called a granum. The plural of granum is grana. And the stroma is the dense fluid inside the chloroplast, which we used to call matrix in the mitochondrion. And the stroma is, of course, very important because it contains the enzymes that are necessary for the light-independent reactions, or the reactions that are not directly dependent on the presence of sunlight. Whereas in the thylakoids or in the grana, we have all the pigments that are necessary for absorption of light energy, and therefore they are necessary for the light-dependent reactions. The thylakoids contain a group of colored pigments, and these colored pigments are grouped into two groups of pigments that are called photosystem 1 and photosystem 2, and they are called systems because they consist of groups of pigments, and photo because they can absorb light energy and therefore be excited, and their electrons will jump to higher levels and will initiate the light-dependent reactions. The photosynthetic pigments inside the thylakoids, together with the enzymes, some of the enzymes lie inside the thylakoids and other enzymes lie in the stroma. They are responsible for carrying out the whole process of photosynthesis. As I've just said, the light-dependent reactions that are initiated by light and that necessitate the presence of light to start occur in the grana, whereas the light-independent reactions that are grouped together into a biochemical pathway called the Calvin cycle will occur outside the granum in the stroma. So here again, if we want to visualize the site of the photosynthetic reactions, then we will have the light-dependent reactions inside the granum and the Calvin cycle, or the light-independent reactions outside the granum. This diagram tries to summarize the processes together. So light energy falls on the colored pigments inside the granum, And meanwhile, there is a splitting of the water molecules. And this splitting, we will be talking about later on, is called photolysis, because photo means light and lysis means breakdown. So water will be broken down into H2 and O. And this O, the atom that is released from water, will be converted later on into molecular oxygen when two oxygen atoms from two oxygen molecules unite together, from two water molecules unite together to form an oxygen molecule. So to summarize, we will be, of course, seeing this in detail later on. The oxygen that is released in photosynthesis comes from water. And then at the end of the light-dependent reactions that start by the falling of the light energy onto the chlorophyll and the photolysis or the breakdown of water and release of oxygen will result in the formation of two important products, and we will be seeing how. The two important products are ATP and NADPH. NADP is the carrier, the electron carrier, and this is one important difference between photosynthesis and cellular respiration, that here the carrier is NADP, that carries an additional phosphate group, not only NAD, and this is the reduced form. How are they important? They are important because Calvin cycle cannot be initiated without them. ATP will provide the energy necessary for Calvin cycle, and reduced NADP will be necessary to give the hydrogen. Why do we need hydrogen in Calvin cycle? Hydrogen in Calvin cycle will be needed to reduce carbon dioxide from atmospheric air into the sugar, which is the end product and the target of photosynthesis. So the sugar that is produced in photosynthesis, which is essentially glucose, will be associated with the release of oxygen. But oxygen has been released earlier in the light-dependent reactions, and then sugar will be produced at the end of the light-independent reactions. The link between light-dependent reactions and light-independent reactions is those two important molecules, ATP and reduced NADP. Of course, as you know, when ATP has given its energy to the Calvin cycle, it returns again ADP, and that ADP will be reutilized in the light-dependent reactions, as will be explained later. Similarly, reduced NADP, after having given hydrogen to the Calvin cycle, will return to be reutilized, and it will return in its oxidized form. This diagram is crucial in understanding of photosynthesis because it shows you in a very simplified way all the reactions of photosynthesis and how they are interrelated, the light dependent and the light independent, and how the products of the light dependent reaction will be utilized by Calvin cycle, and how the products released from Calvin cycle will be reutilized by the light dependent reactions. Now we come back to the photosystems that we have mentioned in the slide on the chloroplast. We talked about photosystem 1 and we talked about photosystem 2. Photosystems are molecules or complexes of molecules, I mean a group of molecules that are characterized by being light harvesting, meaning that they collect the energy from the solar light energy that falls on the plants. And where are they present? They are present on the folds of the inner membrane or the thylakoid membranes of the chloroplast. In each thylakoid, of course, the thylakoid is a microscopic structure, so it is a very small structure and it contains only a few hundred of molecules of these light harvesting molecules. Of course, the most important pigment inside light harvesting molecules are all pigmented molecules. it is the pigmented molecule that is capable of absorbing the light energy, and the most important of one of them is the green molecule called chlorophyll A. So that's why in each photosystem we consider chlorophyll A the reaction center. Why do we call it center? Because it is the most important molecule in among all these pigmented molecules. Why do we say reaction center? Because of course these pigmented molecules will carry out reactions. What kind of reactions? They're gonna lose electrons and then receive other electrons and so on, as we will be seeing in the diagram. Um, Around this most important molecule that is called chlorophyll A, we have other pigment molecules that we call antenna pigment molecules. They surround the chlorophyll A molecule or the reaction center, and they help this chlorophyll A molecule to like attract all the energy and absorb it, to try to absorb as much energy as possible. Why do we call them antenna? Because they're very similar to the antennas that we use or in old radios, for example, in the past, like 50 years ago, radio devices had antennas to try to collect radio waves from the environment. Um, these photosystems are grouped into photosystem 1 and or classified into 1 and 2, and this is the order in which they were discovered. This is not the order with which they work inside the chloroplast. This doesn't mean that photosystem 1 works first and then photosystem 2. No, this is the order of the dates on which they were discovered or described. But actually, photosystem 2, the one that is called photosystem 2, will work first, and then it will be followed by photosystem 1. Why are we splitting them into two groups? Because every one of them is capable of absorbing a certain wavelength. For example, photosystem 2 will absorb light in the range of 680 nanometers. Nano means 10 to the power minus 9. That's why photosystem 2 is called photosystem 680. And then photosystem 1, the molecules that, or the complex of molecules that are present in photosystem 1 is called photosystem 700 because the peak absorption of light energy is in the range of 700 nanometers. Here we have the visible spectrum of light. You know, of course, that there are wavelengths of light that we can visualize, and there are wavelengths of light that are too short or too long to be visualized. So, for example, the visible spectrum of light ranges from the wavelength of the visible spectrum of light range from 400 to 700. So, for example, here we have the longest wavelength in the visible spectrum of light is the one for red, which is 700 nanometers. And the shortest one is the one for violet or purple, which is 400 nanometers. And in between them, we have the green range of wavelength that is around 500, and the yellowish, which is around 600. As we said, the unit is nanometer, which is 10 to the power minus 9 of a meter, or 1 over, I would say, 1 billion meter. Now, as you know, that the wavelength here, as you see, is the distance between two peaks of two, in the diagram for a wavelength, or in the diagrammatic representation in this waveform, this is the peak of the first wavelength, and this is the peak of the next wavelength, and the distance in between them is the wavelength. You know that the photons that constitute light energy, the photons have wave properties, and at the same time they have particle properties, and this is what you studied in physics. And particle properties means that they have a mass and they have a velocity. But when the photons hit a certain body, a solid body, they lose their mass, and this mass is converted into energy, and that's why they give us this kind of heat energy and light energy. The visible light is only a small slice of the electromagnetic spectrum, meaning that if you look here at the electromagnetic spectrum, it starts at a wavelength 10 to the power minus 3 nanometers and ends by a wavelength 10 to the power 15 of a nanometer. Of course, you know that the longest wavelength is associated with the lowest frequency, and the shortest wavelength is associated with the highest frequency, as you can see on the diagram. But the visible, this is again the whole electromagnetic spectrum. I mean the whole range of wavelength. But if you look here, you can see that the visible spectrum, VIS stands for visible spectrum, is only a very small part. So all what we see of colors, starting from the purple color to the red color, represents a very small part of the whole electromagnetic spectrum. In this diagram, you can also see that the shortest wavelength, which corresponds to the highest frequency, is for the gamma rays. Gamma rays have a very high frequency, very short wavelength, and therefore very high energy. The highest energy is for the highest frequency for the lowest wavelength. On the other hand, the longest wavelength is associated with the smallest frequency and with the lowest energy. One of the highest energies are associated with the gamma rays. That's why we use the gamma rays in medicine for killing cells, and we use them for cutting tissues. So we have in medicine what we call gamma knives. Gamma knives are not true physical knives, but they are very high energy that can cut into the tissues. You can also say that X-rays that we studied in human biology and that are used to visualize the internal organs, whether they are radio opaque or radiolucent, then they have high energies. Ultraviolet rays of the sun have very high energies. So descendingly, we have the gamma rays, the X-rays, and then the ultraviolet rays. On the other hand, the longest wavelengths are associated with the longest known wavelength for human beings are associated with radio waves and then the microwaves. So the microwaves are shorter than the radio waves, and the infrared rays, we call them infrared because they have longest wavelength. The red is here, and the infrared is after the red, to the right of the red. So it has a longer wavelength than 700 nanometers. On this diagram, we can see something very important, which is called the absorption spectrum of the photosynthetic pigments. As I said before, the photosynthetic pigments have the ability to absorb light energy. And here we are showing you the spectrum or the ranges of the capacity of the photosynthetic pigments to absorb light. So here on this diagram, which has a title of absorption spectrum or spectra of photosynthetic pigments, on the x-axis you have the independent variable, which is the wavelength in nanometers, and on the y-axis you have the absorption, the how much percent of the energy that falls on the pigment is absorbed. So again, the absorption spectrum shows you the capacity of every particular photosynthetic pigment to absorb a certain wavelength. For example, if I look at chlorophyll A is the green curve. Chlorophyll A is the green curve. So we have to study one pigment at a time. as we used to do in any experimental procedure. So we are going to focus on one pigment, which is chlorophyll A. And absorption spectrum means for every wavelength, how much percent of this wavelength will be absorbed by chlorophyll A. And then after having recorded this, we will do the same for chlorophyll B and then for the other pigments. For example, you have lutein, which is a yellow pigment. You have beta carotene, which is an orange pigment. You have lycopene, which is a red pigment. And you have zeaxanthin, which is another yellow pigment. All these pigments are present in different mixtures, in different ratios inside the chloroplast, and that's why the chloroplasts have different colors. But the most abundant one is what we call the reaction center, which is chlorophyll A. So if you look at the absorption spectrum of chlorophyll A, you will find that the peak absorption of chlorophyll A is between 400 and 500, for example, at 440 nanometers. And then it has another peak of absorption, sorry, at 680 nanometers. Of course, since chlorophyll A is the main reaction center, then if chlorophyll A has a major absorption or the peak absorption as 440 and then another peak of absorption at 680, then the overall absorption spectrum of the pigments will be essentially that of chlorophyll A, or depending on the concentration of chlorophyll A, because it is the most abundant in the chloroplast. On the other hand, chlorophyll B has a peak closer to 500. Lycopene, for example, has an absorption spectrum whose peak is at 500 and so on. So the majority of them have their absorption peaks between 400 and 500, and then they have no absorption peaks in the green and yellow area, and then another absorption peak here at the red area. If they don't absorb the green, then they will reflect it. And that's why we see them green. Because when you see that the plants or the leaves are green colored, this is because they reflect the green light. So when they reflect the green light and it falls on your eyes, your brain will interpret this as green. So why do we see the plants green? Because they do not absorb the green color and they reflect the green color. So a color that is whose wavelength is not absorbed will be reflected, and the visibility of it will be dependent on what it reflects, not on what it absorbs. Here we have the absorption spectrum of chlorophyll A, the green one, and then here chlorophyll A is the blue one, chlorophyll B is the green one, and then we have a different range of or a more extended explanation of the absorption spectrum of photosynthetic pigments. Why am I saying this? Because there are certain pigments, like for example, phycocyanin and phycoerythrin, that did not figure on the previous graph. So if you are wondering why there is a second graph, this second graph is more explicit. It shows you the pigments that you saw on the previous graph and then other pigments. So this graph is enough. You can only utilize this one because it contains a broader number of pigments, although here we have certain ones like lutein and beta carotene that did not figure here. We have carotenoids. Carotenoids include the beta carotene. So actually this one is more explicit. So the absorption spectrum of chlorophyll A and chlorophyll B show that the absorption is highest here in the blue range. And this is normal because we do not see the blue color. There are no blue plants. This is because they absorb the blue color, which ranges between 445 and 500, and they also absorb the red spectrum, which is from 620 to 740, and that's why we do not see red plants. The least absorbed, as I said, is green, and that's why we visualize the green, because green is reflected. So the wavelength that are optimally absorbed and used by the green plants, which fall in the blue region and the red region, these are the ones absorbed. These are the ones that are not reflected. And if you reflect on this, if you think about it, you will understand that you might be wondering, why do the plants absorb blue and red mainly? This is most probably because the energy associated with the blue wavelength and the energy associated with the red wavelength, notice that one of them is high energy with high energy with high frequency, which is the blue. The other one is low energy with low frequency, but both together help the process of light-dependent reactions to occur. One of them is important to excite the electrons, give the high energy to excite the electrons, and the other one will complete the work, and we will see how. So photosynthesis is dependent on light harvesting molecules that are called the pigments. The most abundant ones are chlorophylls A and chlorophyll B. We are just repeating ourselves. Chlorophyll pigments predominantly absorb in the blue range of light and in the red range of light. They reflect the green light, and this is why the leaves appear green. There are, of course, other pigments that are present in the leaves, and we can see them. The orange, for example, pigments, the red pigments are not usually visualized on the leaves, but we can see them in autumn when the plant starts wilting, when chlorophylls degrade. Why do chlorophylls degrade? Because in autumn, light energy diminishes, and the synthesis, not the work, not the functioning, the synthesis of the green chlorophyll molecules A and B depends on the presence of light. So if you don't have enough light, if there is dim light, then there is not enough synthesis of chlorophyll. So the green pigments are no more synthesized in higher proportions, and therefore it's the time for the red, yellow, and orange pigments and a mixture of them to be seen on the color leaves. And of course, we all know that wilting leaves are not very green, and they are rather yellowish or having an orange color. We have a procedure that is called paper chromatography, and it is a common technique in chemistry to separate mixtures of molecules. For example, you have like blotting paper, and then you put a drop of a chlorophyll molecule on a chlorophyll solution on the start of the blotting paper, and then the molecules of chlorophyll will start diffusing along the blotting paper. Of course, this is not blotting paper I'm pointing at, but it's the same idea. You dip the blotting paper in a solution of chlorophyll, and then there will be the blotting paper, the fibers and the spaces, because blotting paper is made of cellulose and it has intermolecular spaces, so the molecules will diffuse along. But as they diffuse along the blotting paper, they will be